Hello.
Hello. Hi, Brad.
How are you?
Uh, yeah, I'm fine. I'm fine.
Good.
Uh,
Good to see you here.
yeah,
So why don't I get the meeting started? If other
okay.
people come in, we'll catch them up on what we've been talking about. So, how have you been doing over the last few weeks?
Yeah, things have been okay. Just finished with my NSEM Viva exams. After that, was it was okay, I think. Uh, the New Year's then. Uh, Otherwise, things are still here in lockdown only, right? So
Oh,
restriction, movement is restricted, kind of. So yeah, that is there. Then
oh, that's good. Yeah.
yeah,
Well, thanks for showing up.
yeah.
Yeah. Welcome to the meeting, and we're gonna talk about a couple things this week.
Okay.
This is the first week of 2022, or first meeting of
Yeah,
2022. So, welcome to the new year. We'll talk
yeah.
about some new things.
Yeah, yeah.
Yeah. So let's see. First thing I want to talk about is I can launch right into or Google Summer of Code news, or what we want to do this year.
Okay.
So as people know here, we are affiliated with INCF. That's our
Uh,
managing
Parent
organization.
organization. Yeah. Okay.
Yeah, and that they're the ones who interface with the Google Summer Code program, so they manage everything and sponsor us. So
Okay.
this year the program's going to be quite a bit different. They're going to have two different lengths of program, I think a 10-week and a 22-week program. So I'm not
Okay.
sure about the details of I just got an email this morning about what they want exactly in the calls for involvement. Like when your group puts forward a proposal to the sponsoring org and they advertise it and get people.
Okay.
So that's that. And then we also thought about having our own organization. Oh,
Okay.
hi, Dick.
Oh.
And so our own organization is the idea there is that I've got some other people that are interested in participating, but they don't have an sponsoring organization. So this year, Malin at INCF suggested, that if people want more slots and if they want to do so, they can fill out their own mentoring organization application, and then
Okay.
INCF would sponsor it, and it would be a better chance of us getting accepted that way. The
Okay.
benefit there would be we'd have more slots and we could do
Okay.
more things, but it's up in the air. I mean, probably we'll do both, and then we'll see which one works out.
Okay.
So I have a let's see. So I'm going to share my screen. So let me see where I put this stuff. I think I put it in here. Yeah. So these are the proposals that I've come up with, or we've come up with, and I put together. And so we have two, and I think that's probably enough. I know last year we had
Okay.
three, but this one is, I don't need that one for this document.
You will connect ones.
So the first one is GNNs as developmental networks. So this is graph
Okay.
neural networks. And
Okay.
the idea is that we have all these networks in biology, in developmental biology, neural
Okay.
connectomes, gene regulatory networks, interactome networks, and anatomical networks. And then we can do basic what we're doing with the work in deep learning and with DevoLearn, and we can construct these graph neural networks libraries and fit them into that toolkit. So we have our deep learning toolkit, which is a number of different models that we can use on analyzing data. But then we could also use these graph neural networks. And the benefit here is that we can create graph embeddings that resemble actual biological networks. So we can, I don't know exactly how this would be pulled off, but that's one of the things I like to do, is like to get people a chance to sort of propose their own solution. And then once they do that, you have, say, like 10 applicants. You can find the best one, and then the people who don't get selected can, follow up on that on their own, or depending on how many slots we get, we can accept more people. But this is, so this is just kind of using these kind of graph embeddings to sort of characterize biological rules, look at the differentiation of nodes in a network. So once we have these graph embeddings, we can do a lot of things with them potentially. But we need to get that that work done first. So that's why I propose that we start here. So the goal is to create a library of GNNs that can simulate developmental processes by analyzing time series microscopy data. So we have data that we've been using for our deep learning work. We can just simply use those data sets and maybe find a few more, and that would be that. And so, we do this, we kind of tell them what they can do before the program starts. It's the same as every year. And I'm not sure about the skills and requirements. I may have to update that, but I think PyTorch and TensorFlow, if you want to use one or the other, then that's a good basis for it. Yeah. So that's the first project, and I'm not sure about the GUI. I don't think we need to do that, so I'll do that. And then the second one is digital microsphere, and that is a project that is the one we proposed last year. This is with Susan's data. So this is the one where you have her ball microscope that can take pictures from all these different angles, and you can also use this flipping microscope technique. So you have all these data that basically show an embryo, the surface of an embryo with different perspectives. And then we'd build a spherical atlas of this. So it would be like, you'd be able to tile the surface of a sphere and explore those, that surface. And, so this is basically, I know we had a couple people produce artifacts on this last year. I don't know. I'll probably send it out. Hopefully they'll reapply.
Okay.
But this is another. This is again one of these things where you have to come up with your own solution. Just basically say, I'm going to use this technique. This will work good. With this project, there's a lot of kind of like figuring out how to do these tilings of space. So you take like flat images and you're tiling them onto a sphere. So there's some mathematical transformation that has to happen. And then you have to have a base sphere that you can tile these things onto that you can navigate. So there's a bit of work that goes into that part. So it's a little bit different than the first one because you're not just throwing an set of algorithms at it. There's a lot of stuff that you need to kind of figure out how you would, they're going to be different tools. It's going to be more heterogeneous, I guess. So,
Okay.
And then the skills I think are handling higher dimensional microscopy data, building an intuitive GUI, feature extraction. So yeah, these are the basic skills. And so these are all the ideas. Are there any ideas about, if we want to add another project or if we want to, do you count differentiation tree and phylogenetic trees as networks? Yeah, actually we do. We can count those as networks. And that's something, when I guess we can, in parallel with that project, we can kind of characterize different things that are networks and kind of put together a short list of those things that, how we might work with them in terms of not only as networks, but like in these kind of graph neural networks. So that is a good question. So any thoughts about the projects or I guess other thoughts?
Uh, what could be the potential applications for the graph neural network project? Like, could it be protein synthesis and all those things, or maybe something more basic? Uh, because I remember like AlphaFold was using something similar, to uh, do their protein, like solving their protein folding problem.
Right, right.
Do we have like some examples in mind with this? Like,
Well, I guess you could, there are a couple things that we could do. We could characterize networks just in general. So, we could build
okay,
the embeddings, and then the idea would be you'd be able to train different types of data and get
okay,
analyses. So, with the deep learning stuff, it was just like taking microscopy images of an embryo and segmenting those images, and then using the segmentation information to build these classifications data sets,
okay,
right? So
okay,
then we
okay.
have like numeric data that we can
So
analyze in different ways.
microscopic embryo related data
Yeah,
will be
yeah.
like the okay,
Of course, it could be other types of data as well. It could be like,
okay,
but the idea would be that depending on the type of graph you're interested in extracting, you would look at the
okay,
data and then say, this is how it's going to be extracted and analyzed. These are the
okay.
features that are important.
So
So,
it's very open ended right now. Okay,
yeah, yeah.
okay,
Let me see. I have another message in the chat. See my papers on Google Embryo. Yeah, I think we cited one in the list here to read. So, yeah, this one here. So, this is the Google Embryo for building quantitative understanding of an embryo as it builds itself. And yeah, I usually give people these two papers to read if they're interested in the project. So it's a good starting point. So, yeah, send along the copy because I'm not sure that people can always access it. So. And then just making some notes here on this. So yeah, I think that's good. Yeah, so I think the only benefit of having another project here would be that if we don't get any applicants for these, we'd still have another one. But I think these two will probably. This one will probably get a lot of applicants. This one will get a few, I'm sure. So we'll see how these go. This all starts like we have to have these projects into INCF by the end of the month, and then if we apply for, or we will be applying as an organization, and that'll be another month after that. So by March 1st, it should be the beginning of the application period, and that's when people come and start asking about the projects and then applying. And I work with them mainly to develop the proposal, and then we get some pretty good solutions to some of these things. So we'll see how that goes. I think it'll be a good opportunity, if nothing else. So that's good. Any other comments or questions before we move on? All right. Let's see. Oh, we got one here. Yeah, sent. Thank you. So, speaking of this DevoLearn, this is the link to a preprint here. And I think I showed this a couple meetings ago last year. And this is now, I've got this up on the ResearchGate as a stub. So, this is the DevoLearn paper. So I've had it on GitHub, and I'm not really sure it's long enough to really publish it or even make it into something like an archive preprint. But we got 11 reads on it. I just, yesterday when I looked, we had 3, so that's good. But this is the preprint for DevoLearn. So we were working on this all last year. And I had I've turned it into, what I did was I created, well actually it was, I think it was myself and Mayuk, but we created this document in Markdown, which is the language that we use on GitHub. You make commits to GitHub in this language. It puts together this document. And then I converted this to a PDF using atom.io, which is a nice tool for converting things from Markdown to PDF, or HTML to Markdown, or you can work with LaTeX in it as well. So that's a nice document tool. And I created this PDF. So Mayuk, Mainak, Bradley, and Ujwal are all the authors. And then the abstract is pretty short. We have a summary of the software here. And then there's a little gap here. I can view the file like this. There's a little gap here because the image is... Actually, this image is actually... it's animated in the Markdown version, but it's static here. So that's the only disadvantage. And then, headings about accessibility, statement of need, technical details, and then walking through segmenting a C. elegans embryo. Again, these figures are animated in the Markdown file, so it's with the PDF, it's like limited utility. But that's just something that is the disadvantage of a PDF file. And we have our data sources, generating synthetic images using GANs, hyperparameter optimization, lineage prediction, which is something. So this basically goes through all of the different features of DevoLearn. And I was surprised when I went through this to put this together, how many things there were. And this is from Mayuk and Mainak's projects, as well as some other documentation that we wrote up on it. So, and then the DevoLearn platform, which is this GitHub organization that has the different. So we have DevoWorm AI, we have DevoLearn, and then we have the DevoZoo, which is something we haven't talked about in a while. But this is the collection of data sets that we have for different species. And it's something that I'd like to get back to at some point, but it's, maybe it needs to be upgraded a little bit more. But I think there's there's a solid interest, I think, in people talking about different model organisms. So, we talk about the name of the group here is DevoWorm, but we could be talking about DevoFly or DevoHuman or DevoMouse. Those are all relevant to understanding development on a broader scale. So this DevoZoo is actually a nice resource, and it needs to be developed more, but it's all under this umbrella. And the idea here is to kind of, nice thing about a preprint is that we can add in, if we decide that DevoZoo needs to be highlighted a little bit more in here, we can highlight it a little bit more and describe what it is. In fact, I think that'll be the next version, is going through DevoZoo and even DevoWorm AI, which wasn't really mentioned here too much. But so that's that, and I think that's a nice way to kind of cap off some of the work that we've done on DevoLearn. And I don't know what the next steps are for DevoLearn. I guess it's just as we get people coming in, they can maybe add on their own functionality, or I think the big thing though would be if we could get a user base for this and people actually using it. And but that's something that we haven't really seen. We've had a lot of downloads of the software, but it's it's difficult to really build any more of a community around it, or build any sort of more work if we're just people are just kind of using it and thinking about next steps. So Mainak and Mayuk haven't really been involved with it much lately. By lately, I mean the past 2 or 3 months. So they might come back and work on it some more, or we might get some new people who might want to do some more work on this. So that's very good. This is something I want to keep sort of sustain, and I want to keep bringing it up and coming back to it. Let's see, we have a comment. Suggestion, write people who write papers on model organisms. Yeah, that would be a good idea. Okay, and then yeah, I think that's a good. I know there have been some interesting reviews of model organisms of late. I think we've reviewed a couple in the group, and then there are others that I've seen that I need to go back and grab hold of, because I think that's an interesting kind of topic to build on. And so, work on model organisms is twofold. One is like, if I'm working with one model organism, I always ask the question, how does my work generalize across the tree of life, or other model organisms at least? So people working on, Drosophila or C. elegans, they're typically focused on their own world of that organism. And, people will use, say, like C. elegans to look at things in humans, but it's much better if we could get like a broader characterization of some of these things. And so, sometimes when you look at model organisms, processes are very different across different animal species, or even animals and plants, or however you look at the tree of life. But sometimes they're very similar processes. And so, where you can find those similarities, something like DevoZoo is nice because you have this cross pollination of the different model organisms, kind of put them in a broader context. So look, I'll think about the people who, maybe I'll contact some people and see what they're thinking. Okay. The next thing I wanted to cover is game theory of developmental processes. So this is a paper that I just put this preprint out. This is something that I think I presented last year, a couple months ago in the group. This was something that was submitted to the Dynamics Days conference, which just happened this last weekend. So it was a pretty decent conference. It was hosted by Georgia Tech, and they were, virtual because they, couldn't meet in person. And so they had the whole thing set up. They had talks, posters. We got in as a poster, so this is like a 2-minute thing, and that video of that presentation is on YouTube, on the YouTube channel. So if you want to see that poster, it was a very short 2-minute summary of the poster. So then this preprint is meant to be a supplement to that poster. So it's like, you want to find out more, read the preprint. So that's why this is here. Actually, I just wanted to write up a preprint for it to keep the idea moving forward. But there's a longer 30-minute talk also on the YouTube channel for this basic idea that I presented at another Dynamics Days in Europe last year. So, and it was also virtual. So this is the preprint, and it actually goes into a lot more depth in the different presentations. So let me view the file here, walk through this a bit. So this is a paper that takes developmental processes and applies game theory models to it. So the idea is that you can use this class of model to model developmental processes in different ways. And there are different types of games, and there are a lot of different types of developmental processes. So there's a lot of variation there that but that the general idea is what I just said. And it also requires looking at game theory a little bit differently. So people have thought game theory started back in the 40s and 50s with John Nash and von Neumann and some other people. And they came up with these basic models of game theory. It's basically, games of strategy. So something like they call the tit for tat game, which is a game where you have one player plays a strategy and another player tries to one-up that strategy. And then the other player responds by one-upping that strategy, and so on and so forth. There are other strategic games like chess and poker, and those are all, you can model those as games, so you can basically use a computational model to model those things as games. And this was something if you take the chess example, this was something that was used in early AI to sort of serve as a model for intelligent behavior. So an AI would play chess. It would be able to play like a game of chess, or maybe like a game of tic-tac-toe, or what's called in the paper here, noughts and crosses, or something similar. And it would just implement basic strategy. It was usually some combinatorial game because computers are good at combinatorics. And eventually this became something that was able to beat humans. So in the 90s, IBM's Deep Blue was able to beat a chess grandmaster. And so that wasn't a long time. That was like 40 years. But anyways, that was the idea. So we can use these for developmental processes. And they're different types of games. But before we get to that, we have to talk about different types of things that the agent or the player has to embody as a developmental agent versus like a strategic human player or something like that. The idea of rational strategic player is a little misleading because although it's been applied a lot to humans, they've also used this to analyze animal behavior. So you've had games that can model like birds and their visual behaviors and their different decisions that they make. But still, even in birds, you have like a cognitive agent. It's a cognitive agent. In developmental game theory, there's no cognition assumed, because you could be dealing with an organism, you could be dealing with a cell, you could be dealing with molecular interactions. So there's this idea of the ontogenetic agent, which is this type of agent that doesn't explicitly have cognition, and it doesn't behave in this rational manner. It behaves sort of in a natural manner. And so what that means is that you have these, it basically operates on the principle of fitness. So this is a lot like what they call evolutionary game theory, where the agents are evolutionary, they have a fitness, and they sort of maximize their fitness for each of them, and then that leads them to these different evolutionary trajectories that end up, either in some equilibrium or some other result that is able to maximize fitness for each player in the game. So the other benefit about taking cues from evolutionary game theory is that you have this dynamical trajectory. So in development, of course, that's very important, where you have this trajectory of what goes on in development. And there are actually two ways to view this. One is through an evolutionary game theory lens, which is maximizing fitness. But there are also these iterative games, which are forms of games that get played over and over. So I think there's one example in the paper of like the prisoner's dilemma, where there's a form of the prisoner's dilemma, which is the iterative prisoner's dilemma. And so you have this single game that you can characterize. And then you have the iterative game, which can just be played over and over. And the payoffs change based on the past states. So the past state informs the present state, which informs the future state. I'm trying to find that table. Maybe I don't even have it in here. But there's a table that kind of goes over the prisoner's dilemma and its payoffs. I don't have it in here. I don't think I put that one in. Anyways, the prisoner's dilemma is a game where you have two prisoners, and the idea is that they're going to, you have them in different rooms, and the idea is you want them to confess to a crime. But if they can coordinate their behavior, they can both sort of not confess. You threaten them maybe that the other person is talking right now, or that you're going to punish them severely. And so the idea is that to reach a optimal equilibrium for the prisoners, they both cannot say anything that would incriminate each other. But oftentimes they'll incriminate one or the other, and so that's where there's a suboptimality there. So that's the prisoner's dilemma, without going into too much detail. But the idea then would be, with the prisoner's dilemma, the iterative version, do both prisoners, are they able to coordinate the information that they have over a long period of time, or do they just do it once and then it breaks down? And so, there are all sorts of games that I feature in here. There's a 0 order or zero player game, which is a set of games that involve like different point processes, and the game of life is actually considered a zero player game. Then you have single player games, which are games against nature. And this is where you have some natural process, a stochastic process, or it could be deterministic, which isn't very fun, but like the stochastic process. And then you have an agent that's playing against that stochastic process. So the agent's trying to predict what the stochastic process will do. And of course, being a stochastic process, that's hard to predict, but the agent will develop some sort of strategy for that. And then there's a payoff. If the agent is correct, they get a payoff of 1. If they're, yeah, actually, if there's a, I think if they, I can't remember how this works, but there's a payoff that they get for some sort of correct choice. And then for a neutral choice, which would be like something that's not correct, but something that doesn't hurt the agent, it's a 0. And then if it's something wrong that hurts the agent, it's a -1. So then you can add this payoff over time. So this collective payoff is, there's an average payoff, a cumulative payoff. You can calculate it in different ways. And then you can say, I see why that process unfolded the way it did. Because that payoff is, there's maybe there's some mechanism in the agent that's able to predict something in nature, but it only is able to do it sporadically. So you can kind of understand things like that. And that's actually quite valuable at the molecular scale, where there are a lot of processes that involve like recognition. But, like say, for example, cells, when you look at like things like translation or binding sites, things like that, it's a process where there's a lot of this going on, and not all of it actually unfolds in the same way. Sometimes, there's just a lot of stuff in the cell that needs to be bound, and if enough of it is bound, you get this activation. So not every agent is going to have a clear payoff, and so that's the idea behind this. And then there are these the n-player games, which are like 2 or greater number of players. And there are a number of examples from behavioral biology and other types of rational agent behaviors that we can use and apply to this developmental context. So in this case, there were 2 examples: the prisoner's dilemma, iterated prisoner's dilemma, which I described previously. Then there's this noughts and crosses, which are the first mover games. And this is something we published a couple papers on. We published a paper on this in embryos, which is in this paper, Origin of the Embryo, that was done a couple years ago, where one of my contributions to that was to put in one of these first player games. So the idea is that you have these different cell lineages that they differentiate and then they divide. And the idea is they're in this confined space, and the lineage that divides first is the first mover, and then the one that divides second is the second mover. And the first mover in general constrains the second mover's behavior. So if the first mover divides, and they have more space to expand, the second mover then can divide and expand, and then so on and so forth. So this might explain timing in a lineage tree. You could also apply this to the connectome. And so how does the connectome get assembled? And I actually used some data from C. elegans, both cell differentiation data and synaptic data, to show how this connectome assembles, taking this principle of first mover dynamics and deriving some rules or some scenarios where the data fit in. So the data get classified by these different rules, and they fit these strategies. So then there are other types of things like systems, equilibrium, and complexity. So there are different types of combinatorial games, evolutionary, more explicit evolutionary games, and what we call spatial games, which are things that look at space as a set of potential strategies. So, agents will obtain a specific spatial structure as a collective strategy to defend against invaders. There are other things like that. And then there are the broader systems level phenomena, which I go through. So that's that paper. And I like the idea, particularly of natural agents, but some of the games, I mean, this is like a preprint, so we can expand out. We can come up with new games or kind of discuss more about some of these things. I was thinking even of what we might call an Evo-Devo game, which is where we apply evolutionary game theory more explicitly to the developmental context and look at how that maybe explains some things we see in the evolution of development. A couple of, okay, so suggestion, right people, okay, that one. Only 932 papers in Web of Science. Was that for, oh, that was for model organisms, or?
Yes.
Okay, thank you. Yeah. Could extract email addresses. So maybe, yeah, look in Web of Science and see what they have, if we have some interesting things there. And then differentiation wave is a simple purpose. Okay. Did you want to expand on that? Yeah, I mean, it's this part here, the part about like, you know, purpose and natural agents. It's an interesting topic. Oh, the stone papers, yeah. So yeah, I have to look at that a little bit more. I can't remember exactly. Even the paper, the origin of the embryo that I was involved in, I can't remember. It's like you get to a point where you can't remember. You remember kind of what every paper is about, but like kind of, Bernays has to remind you. I'm sure Dick knows that. It's like, anyways, so that's good. Any comments or questions at this point? Okay, so next thing I wanted to move on to was a couple things for 2022. We had a couple things that kind of ended at the end of the year. We had the submissions document and the major tasks, which haven't been updated, you know, as regularly as I would like, but that's okay. So this is our, like last year, this is our submissions list. So at the beginning of 2021, we started this list, and we had 34 things in it from last year. And this is like things that were proposed or that we did for different venues. Some things never got anywhere, and some things got accepted and presented. Some things were rejected. And so, you know, there are all these possibilities, but we wanted to assemble them in this document. So this was for 2021. And maybe I had a comment on this once. I had it open, and someone said, "Is that something you're going to go through? Because this looks pretty illegible." Which, I mean, it is. If you look at it like this, we usually zoom in on a couple lines, but anyways, maybe doing this for 2022 and starting off with some of the new submissions. So 2022, and this is 2021. So let's go to 2021 here. Okay. So we had a lot of things, and some of these things will carry over to 2022. And so that's something that, so the neuro-IPS was something that was, I think people didn't, I don't think anyone here sent anything to them, but I think that's something that a couple people, especially in my other group, participated in. And so there, and same with NeuroMatch. We had a NeuroMatch, we had some participation in NeuroMatch as well. So that's good. And then this Dynamics Days 2022, which was green, that was presented. So that's pretty good. All right, so we have some things here, some of these items that never, we didn't really do as much with in 2021 as we would have liked. Maybe we'll get some of these and pull these over. These submissions, these are the ones on the test of Williamson's, what is this one? Williamson's symbiosis. this is the idea of, well, it's an old idea that we've talked about in the past. Oh, I can't add a row here. Anyways, maybe we'll do this later. Oh, no, here we go. Okay, so let me get this. So we have that one. We have a couple others that involve diatom movement. And this is something I know I presented some stuff a few weeks ago on that. So that might go somewhere this year. This Steve McGrew's book, Eye of Nature, I still need to go through that and see what we can do with it. We talked about publishing it or, building upon it in some way. And then this quantitative comparison of archaea and shaped droplets, which is still, I haven't heard from my knock on that. I still wanted to get in touch with them about the presenting on it, but I haven't heard back. So we'll see, present in the main meeting here. I know Susan presented, I think it was last meeting that we had of 2021, and that was a very good talk, and that was on some of her work on the biophysics of well, I can't remember exactly what the title was, but it was a pretty good talk, and we had there's some follow-up from that as well. Okay, so add one-dimensional Ising is Wolfram pattern generator. So that's a good one. Let's see, this one here. I'll fix the formatting later. And then line 26 Basileria, which is, I guess, line 26 from 2021. And that's this one here. Oh no, this one. Yeah, smoother jerky. So that's already in the list. So we have that in the list, 2022 list. We have some, yeah, so this is the different diatom things. And then the one-dimensional Ising is Wolfram pattern generator. Yeah, there's some interesting things going on with pattern generation. In terms of both, kind of thinking about how it's generated and then looking at some of the outputs of that visualization. So again, this is something that, this list is just very basic ideas. We can go over this when we want and kind of, figure out what we want to do with it and where we want to submit things and so forth. So that's good. Let's see. Yep. No, I don't want to do that. Okay, I'll fix that later. The other thing is major tasks for 2020, 2021, which I guess now is 2020 to 2022, so I'll change that later. And so we have all these cards here. And I didn't want to have to go through them point by point, but I just wanted people to be aware that these are still here. And so some of these things are finished. I'm not logged in so that's okay. We'll do that later. But some of these are finished. Some of these two are finished, for example, or at least partially finished. I don't want to say we're not going to revisit that again. It's just that, for now it's finished. A lot of things here to do, to possible things. So this kind of ties into this submissions document where we kind of plan things out. So if you're looking for something to do, a way to contribute, these are things that we can revisit and kind of make kind of find something to do. We have a lot of things that we've talked about, and this is the way to carry over some of those ideas and align them with people's interests. Okay, fish ladder is submitted. Good. I'll go back and modify this later. Okay. So the last thing I want to talk about today, this is I don't need that. Okay. So the last thing I want to talk about today is getting into some papers, and I know I talked about some things already, but let's see. I had some interesting things to come up. I know Dick has sent me some papers in the over the break, so thank you for that. And so one of the first thing I want to talk about is this microscopy folder. I have a couple interesting papers in here to go over, and following up on our microscopy interest in the group. The first paper is from BMC Bioinformatics. This is quick PIV, it's the technique name. And so this is efficient 3D particle image velocimetry software applied to quantifying cellular migration during embryogenesis. So this is velocimetry software. This is on, I guess, on a data set, and they're actually developing the software around it and trying to get to sort of quantify cellular migration. So in this case, you're not just extracting, segmenting parts of cells, you're tracking them as they move in movies. So it's a series of images, you're tracking the movie. So the background here is the technical development of imaging techniques in life sciences has enabled the three-dimensional recording of living samples at increasingly temporal resolutions, meaning that you can not only capture the movement as like a movie, but you can capture that movie in smaller and smaller time, or the sampling rate gets
Bradley, are
higher
you
and higher.
trying to speak something? Because I think you're muted right now. Are you describing something?
Okay, I was muted.
Yeah, okay.
Thank you. Okay, no sound. Okay. Yeah. So let me go back to this. So this is the technical development of imaging techniques. So the idea is that you have these videos, but they also have a higher sampling rate that you can have. And we've talked about this before. And so there are some machine learning techniques that allow us to take like series of images, time series of images that are sampled at a fairly low sampling rate and increase the sampling rate basically. The idea is that the algorithm will take the images in sequence and then infer the images in between. And so in a normal time series analysis, you can only do so much of that before you experience this aliasing effect. And so, and I don't know what the rule is in machine learning, but suffice it to say, lower sampling rates will not give you the data that you need to infer higher sampling rates or more continuous action, unless you really kind of work at it. And I don't know, but anyways, this paper is a dynamic 3D data sets of developing organisms. So they have these. They've collected the data especially for this. They call it dynamic 3D. Allow for time-resolved quantitative analysis of morphogenetic changes in 3 dimensions. So they have they have a 3-dimensional data set where they collect data from like 3 spatial dimensions and then integrate that and then do the analysis on that. So this generates terabytes of image data, so it's a lot of data. Particle image velocimetry, I'm having trouble with that word today, PIV, as a robust and segmentation-free technique. So this does not involve segmentation, this particular technique. Suitable for quantifying collective cellular migration on data sets with differing labeling schemes. This paper presents the implementation of an efficient 3D PIV package using Julia, which is a nice scientific programming language that in my other group we've talked about it around the edges, but it's a very nice language. It's not Python, but it's something that's really enables a lot of scientific computing. And so this package is called Quick PIV. Our software is focused on optimizing CPU performance and ensuring the robustness of the analysis. So this Quick PIV is 3 times faster than the Python implementation hosted in OpenPIV. So this Quick PIV uses Julia. And so our software is also faster than the fastest 2D PIV package in OpenPIV written in C++. By applying this to the 3D data sets of the embryogenesis of Tribolium canastium, we obtain vector fields that recapitulate the migration movements of gastrulation, which is a phase of development in these embryos, both in nuclear and actin-labeled embryos. So they have, again, a lot of the cell tracking that we do ultimately involves cells that have something labeled in the cell. So it could be the nucleus, it could be actin, it could be something in the cell that you can use as a marker. And you can hook onto that to segment them. But in this case, they're just using these labels as a way to sort of orient their analysis. Now, when I say label, I mean like they do this in a molecular way. They use some sort of fluorescent protein or some sort of antibody stain that binds to something and makes it fluoresce. So it's like this nice fluorescent contrast in the image. And so they're able to detect translations and non-segmentable biological image data. So there are some of our images. We've chosen some of the data sets that we have because they're easy to segment. But in this case, they're not doing segmentation. They're doing this kind of just using these different labels and tracking them. And so they don't have to segment the data here. And they're able to detect things in 3 dimensions, spatial features, as you will. So that's good. So let's see if they have anything about this embryo. Let's see what it looks like. Let's see. Sometimes they put the images towards the bottom. I don't know if they have them. I hope that, here we go. So this is pictures of the embryos. So this is PIV analysis were performed on the actin signal of a double hemizygous transgenic embryo. Before and during, so before is the top, during is the bottom of gastrulation. So this is A, and this is before, and this is during gastrulation. So as you can see, there's this gastrulation process where you get this sort of folding of the tissue. And you can see it's picking it out in this area here. So this is the image that's being analyzed. It's kind of, giving us differences in this. You have the arrows here, so it's showing the movement of cells. And these arrows are, you can see a difference here between the 2 samples. So that's what it's doing. It's picking up these labels and it's seeing how they move in the image, in the video images. And then it's creating these arrows that show the direction and the extent of the movement. So you can see that it has this, there's a difference here in the different samples. And then, of course, you can create numbers out of this and actually do some more analysis on it. And then PIV is also performed for the same time points on the nuclear signal in B. And then the similarity selective averaged vector fields are shown next to the actin vector fields, which I'm not sure what the difference is here, but you can see that you have differences again in the embryo and then these vector fields that result. So, okay, so that's good. So, yeah, this looks pretty good. This is a nice, and then they show this graph here and see the orientation similarity between each pair of vectors and the 2 channels is computed through their normalized dot product. So this is the Euclidean error here and the normalized dot product, and it kind of shows you this in the plot. And then 3 patterns of cell migration can be distinguished in this data set. So the first is segmentable and trackable. Okay, and then the second is segmentable and non-trackable, which is this one, SNT. And then non-segmentable and non-trackable nuclei. So these are classes of nuclei. So you have a segmentable and trackable nuclei, meaning you could both track and segment these nuclei, meaning that they're pretty obvious to the algorithm and they're pretty easy to sort of segment and understand as a little dot. Segmentable and non-trackable means that you can segment these images here, but it's hard to track them over time. And then non-segmentable, non-trackable are these nuclei that you can do neither, and as you can see, they're pretty, you can't really make them out. You can make them out with the eye maybe, but not with an algorithm. So that's what we have when we're dealing with this kind of data. So I think this is an interesting paper. The other one I want to talk about is this label-free 3-dimensional imaging of C. elegans with visible optical coherence microscopy. So I think Susan talked about this technique in her talk last year. Optical coherence microscopy is this OCM method. And so fast label-free high-resolution 3-dimensional imaging platforms are crucial for high-throughput in vivo time-lapse studies. So this means that we want to look at these things live, and then want to look at a lot of data, and we want to look at them over time as a time lapse. And so this is the kind of thing you want to do for C. elegans, because C. elegans has a nice mode of development where you can do this, and it's easy to analyze and easy to understand what you're getting. So despite the needs, methods combining all these characteristics have been lacking. In this case, they're presenting this label-free imaging technique for three-dimensional data sets, sub-micrometer spatial resolution, and it's something they call visible optical coherence microscopy, or VIS-OCM. So VIS-OCM is a versatile optical imaging method which we introduced recently for tomography of cell cultures and tissue samples. So Susan mentioned it as OCT, or optical coherence tomography. There's, of course, a larger class, which is just the general microscopy, but it's the same thing, more or less.
It's imaging with infrared light.
Oh, good. Oh, hi, Susan. How are you?
Into the depth of tissue. Yeah, sorry I'm late.
Oh, good. No problem. Yeah, so this is, so our method is based on Fourier domain optical coherence tomography, and then this kind of tells you what that is. By operating in the invisible wavelength range and using a high NA objective, VIS-OCM attains a lateral and axial resolutions below 1 micrometer. So additionally, we use a Bessel illumination offering an extended depth of 40 microns, which is micrometers. People call it microns as well. We demonstrate the VIS-OCM image properties allow rapid imaging for full-size living C. elegans down to the subcellular level. So that means that we can look at some pictures, perhaps. And if we go into the pictures, this is the setup here. So this is the broadband light source, and they've characterized the intensity. And then this is the sample mounted on a plate, so they have these cell cultures that they look at. And then this is the setup here, the illumination detection, and then this is the spectrometer that they use for this. They use this sort of recorded spectrum with a subtracted background, and then you get this profile. Is there anything you want to say about this, Susan?
No, I haven't gotten into optical coherence tomography like I want to.
Oh, okay.
It's just that it's a tomography. You do depth images, and it can go to 2 to 3 millimeters depending on how you set it up. Usually they say 2 millimeters depth,
Yeah.
so this is a little shallower. I don't know.
Are you familiar with the different types of light source you might use for it, or like?
Yeah, there's one that's spectrometry, which you use a broadband light source, and then there's a swept source where you break out the different frequencies, and you more or less get the same type of image, or it's more or less the same technique, except that one's a broadband light source and one is sort of incremental. So you're separating out the bands.
Okay.
And then you do similar processing with the different frequencies.
Oh, good.
For post-processing, yeah.
Yeah. So you see the step, the Fourier transform here. Karan, did you have? Oh.
Definitely need the Fourier transforms to work with this, and some interesting tensor math that does sparse sets, which I haven't looked into either. Very advanced linear algebra. Hurrah!
Yeah. Yeah. That's good. I think Dick had to leave, and I had a comment from him in the chat. We were talking about the PIV method before. He said a problem, real PIV of flow around a moving diatom. So PIV was this other technique where, I know Susan missed it, but it's this technique where you can look at different things, like different markers in say like an embryo or a cell culture, and you can basically track them over time and get a flow map, which is like a map of arrows. And what Dick is saying here in the chat is that you could create a PIV of flow around a moving diatom, you could use like, some sort of maybe a tracking dye or maybe some sort of fluorescent tracking technique to look at the flows around the diatom itself. So instead of using something in the cell and making it fluoresce, you could use something around it. I think that's an interesting point because this PIV technique looks kind of interesting, and of course we have the computational tools to complement. So
yeah, it's very hard to find a dye that works at the infrared frequencies that do optical coherence tomography. Light tends to go. That type of light tends to go through air and water.
Yeah.
Readily. So finding a dye that works at those frequencies is difficult. They're more or less water windows. Dr. Sharif wouldn't like this, but I'm not remembering exact frequencies. They're 870 nanometers, 1100 nanometers, and 1300 and something nanometers. And some of them are used simply because communication systems are at those frequencies, and the hardware for that is readily available.
Yeah.
Anyway, that's sort of my extent of the knowledge here.
Yeah. That's fine. Yeah. So that's the image there, and I don't know if there are any other images. Sometimes they have an image of the. Okay, here we go. 3D volume rendered images of cut sections of a young adult wild-type C. elegans. So this is our adult C. elegans. This is where it's not a mutant, it's a wild type. And you can see that they've characterized the entire worm from head to tail. This is what a worm looks like under a microscope. You get it's a model, so it's not like doesn't show a lot of the features on the outside here. But it basically looks like this, and you have a cross section here, so you're able to walk through the cellular structure in D and E. So you can see that there's this cross section with sort of cell bodies all along this axis. You have different. This F is a transverse section, which is where you cut it from top to bottom, dorsal to ventral, and you can see this sort of section where you have cells are organized, cut in that way. And so B and C are what? B and C are sections along the x-axis. So those are just sections along this way from head to tail, and you can see the structure in there. So this technique will slice up this C. elegans in a number of ways that will allow you to see the different anatomical views and walk through them. As you know, if you've done microscopy or even fMRI, which is a technique for looking at anatomy, you get these slices. It walks through the slices. You can explore through the slices, and it looks like you're moving through some organ or some organism, from one side to the other, and you have these discrete slices. But in this case, they've been able to characterize all of this pretty well. And this is at a scale of 100 microns here, 110 microns, and 63 microns. So this is this little cube here would be these dimensions. So along the surface of a C. elegans, it might be like a couple of millimeters long, and then the width is not even 110 microns, and then the height is about 60 microns, give or take. And so that's the spatial scale we're looking at. So it's pretty fine-grained, and you can see that it's coming up with some pretty nice images.
Major advantage of this technique is that you can do histological imaging without harming the animal. Like, they're live, and I'd already said that, but it also doesn't tend to harm things. I've done axolotl eggs at early stage, and the axolotl eggs have hatched, and in the case of the female that hatched, she had her own eggs and hatched babies that were normal.
Yeah.
I've already tried this, or my friend Elizabeth has, and it didn't seem to harm them at all.
Yeah. So, like one of the alternatives for C. elegans is something called cryo-EM, which is where they freeze the sample and then they go through and they get these really nice images. The problem, of course, is you have to kill C. elegans to do that. You freeze it, in liquid nitrogen, I believe. So this is probably an advantage here as well. And so this is the pharynx, the intestine. So these are just different parts of the worm, and you can see it's pretty high resolution, where you get you can make out the vulva and oocytes, germ cells. I mean, these are things you would see under maybe a more a normal microscope, but getting this is a little bit higher resolution, I think, than normally you would normally get in some cases. So yeah, they're able to do a 3D model of this. So they're able to pull this together and build yeah. So this is just a benchmark, I guess. This isn't like anything where they're trying to discover something, but this is something that they're able to build a 3D model of C. elegans using high contrast down to the subcellular level, where you can actually identify things inside the cells. And yeah, I think that's good. So that's our microscopy. And then I wanted to talk about one more thing before we go. I don't know if I brought it up here today, but I'm not sure if we want to get into it. Well, I don't think I included it today. But I think I might talk about maybe a physics paper here before we go. So this is an interesting paper, scale-dependent irreversibility in living matter. So we talked about active matter last time, and this is about living matter. So let's get in a little bit into this. I just wanted to go over it a little bit before we go. So the abstract says a defining feature of living matter is the ability to harness energy to self-organize multi-scale structures whose functions are facilitated by irreversible non-equilibrium dynamics. While progress has been made in elucidating the underlying principles, what remains unclear is the role that thermodynamics plays in shaping these structures and their ensuing functions. So here we unravel how a fundamentally thermodynamic connection between the physical energy dissipation sustaining non-equilibrium system and a measure of statistical irreversibility, or the arrow of time, can provide quantitative insight into the mechanisms of non-equilibrium activity across scales. So a non-equilibrium system might be an organism. This statistical irreversibility is the arrow of time. So the idea is that you have this organism or this embryo that's developing, or this organism that's living, and time is basically entropy acting upon the system. It's either developing and growing, or it's sustaining itself, or it's dying and falling apart. And this involves energy flows. So you need a lot of energy when you're growing or sustaining, and energy is dissipated when you're dying and you don't have any incoming energy. It's a very abstract way to look at it, but so they want to understand this mechanism across scales. And so in an organism, it would be like the cells, or it could be the DNA, or it could be like other organelles in the cells. All these things are driven by the same process. Specifically, we introduce a multi-scale irreversibility metric, demonstrate how it could be used to extract model-independent estimates of dissipative timescales. So they use this metric of looking at things that are irreversible with respect to the flow of time, demonstrate how it could be used to extract these estimates of dissipative timescales. What is dissipative just means it's, there's an energy flow there. Using this metric, we measure the dissipation timescale of a multicellular structure, the actomyosin cortex, which is a thing with muscle. This is a muscle thing that involves muscle building and things like that. And further observe that the irreversibility metric maintains a monotonic relationship with the underlying biological non-equilibrium activity. Additionally, the irreversibility metric can detect shifts in the dissipative timescales when we introduce spatiotemporal patterns of biochemical signaling proteins upstream of actomyosin activation. Our experimental measurements are complemented by a theoretical analysis of a generic class of non-equilibrium dynamics, which is they'll probably use something like differential equations to look at the behavior of this over time, elucidating how dissipative timescales manifest in multi-scale reversibility.
Have they included gravity in this anywhere?
I don't know. It's a good question. I think they're interested largely in the energy flows, but that's interesting.
Microgravity and gravity and it's another component of this.
Yeah. So I don't know how this is interesting. Like some of these models of like entropy, they don't really consider like things like biomechanics or other types of things. So I don't know how they're looking at this. I don't think they're considering gravity, I don't know how they treat gravity in it.
No, it's just, I was just thinking about that.
Yeah.
The other day, and how these reactions react differently in microgravity, for instance. Yeah.
Right.
So it fits in with this topic anyway.
Right. I don't know. Yeah, and actually something maybe to follow up on. I don't know how people are thinking about this. Like especially like we've talked about like active materials, and I don't know like what the state of that is, if people really consider things like gravity in those models or not, but or a differential behavior of gravity. Usually I think maybe they just assume a specific gravity. So
right, it's just that I'd like to see them include it. I know some people that did a model of chemical interactions, and I asked them, well, did you include gravity? And they said, why?
Yeah.
Like, really? Well, I tell you what, it makes a difference.
Yeah, I would imagine, yeah. And especially especially if you're considering differences in gravity. I mean, people probably just assume like 1G, which is, what you have on Earth, and that's that. And it's like a vacuum, It's like the whole joke about the spherical cow in a vacuum, or spherical something in a vacuum. That's like the standard physics model. Yeah. So, I mean, a lot of this is like, they're modeling these type of molecular biological systems. So looking at the energetic efficiency of molecular motors, for example, and it's just kind of looking at the different forces, but again, like, they're not considering gravity necessarily.
Yeah, but they're, the molecular motors will operate differently in microgravity. Microtubules don't form in the same way. There's no convection in microgravity, so that complicates a microtubule's life, shall we say. They don't get convection to help with the replenishing of the plus end.
Right.
That's the way I see this, but it makes a difference.
Yeah.
A huge difference. Starting on a small scale, leading up to astronauts having a bad time in microgravity.
Right. So a lot of times they consider, like with these models, they also consider like just a closed system. So like, it's like you kind of fix the parameters and you see what it behaves like under a closed system. But a lot of life is actually an open system, which is kind of an interesting interesting thing. I don't know how you model equilibrium or you model thermodynamics as an open system, but it does make so any non-equilibrium process is accompanied by the irrevocable loss of energy to its environment, often in the form of heat, which commensuratively produces entropy in the environment. So again, this isn't a closed system. Now, when you have, obviously you're losing, you're generating entropy as as an organism behaves, or as it doesn't replenish it can replenish its energy, but you're still losing this entropy. So it's an inconvenient way to think about, like, this energy balance, is to think about it as a closed system. But in an open system, I mean, you still get entropy, but then there's this question of, well, what's, how is a system able to grow and generate things and do things that's not, that sort of, there's an entropy loss, but there's also a lot of energy coming in. So it's one thing I don't really know how I know they're probably thinking about it as a closed system and kind of like isolating the mechanism, but there's a lot more going on there that we can consider. And so again, we have our equations here for thermodynamics, and so actin myosin cortex is a multi-scale dissipative structure. That's their main argument here, and so this is something they were able to measure, and this is the actin myosin cortex. This is these little myosin molecules here. So, and you can see that they've got different positional fluctuations and position increment autocorrelation functions of these endogenous cortical granules, and they're able to probe this for force fluctuations in the cortex. So this is the actomyosin cortex. These are the molecules here, and they're doing different perturbations to it. They're the granules here, the F-actin molecules, the myosin molecules.
What is a granule?
I don't know exactly, but that's these balls in here. I guess they're just like embedded within. So if you see in this image, the red are the molecules. These white things, I guess, are the granules.
Yeah, well, it's a movement of them, but what do they mean by granule? That's my question.
Yeah, I don't know.
Oh, well.
Yeah. Yeah, it's good. You have to dig into the paper for this. But
Okay. Yeah, well,
yeah.
I can look at it. I captured its title, so I'm good.
Yeah. So this, and then this is time irreversibility reveals the dissipative timescales and levels of non-equilibrium activity in pattern cortex. So you can see that they're looking at these dissipative timescales. They're looking at the fluctuations in the intensity here, looking at frequency analysis, and then lag time, which is D, which is this estimated irreversibility of this parameter for 5 different stages as a function of lag time. Inset here shows the time scale of the peak irreversibility, peak irreversibility is a function of row oscillation period. So this is something that they've derived out of this graph, and this is the oscillation period S irreversibility timescale. It goes up as the row oscillation period increases. So it's an interesting paper. I don't want to get too much. I probably spend like an hour on it. It's not, but it's an interesting way to look at the different problems. And I was looking for another thing that I was going to show, but I couldn't find it. But next week I'll probably show that. Anyways.
Yeah, I'm sorry I'm late. I was late. What was the first paper you went through? Or?
It was that imaging paper. It was the, I can send them along. It was this PIV technique, which is like a technique for imaging different, like you image different fluorescent markers, and you don't segment the cells. You just track the fluorescent markers in a cell over time. So you have these movies, and then you can track them. You can create vector fields, which allow you to get a handle on movement in collective movement. And they looked at an embryo actually, so that was a nice way to do it.
Okay.
And, Okay. Well, everyone, thanks for attending our first meeting of the year. I think it was pretty successful. If you have any papers you want us to present in the meeting, you can do that. You can present it yourself if you want. You can also present on different topics. I know there's a lot of stuff that we talk about every week, but one of the things about this is we'd like to see people present maybe a short thing that if they have a topic they want to really kind of explore. Besides just kind of presenting papers like the way we do, we can kind of lay them out a little bit more. So.
Yeah, well, I'm working on a paper right now.
Yeah.
After I get it finished, I could do a short version of it or something for you.
Good, yeah.
Yeah.
And yeah, we'll also be in touch with respect to Google Summer of Code. We're going to be working on that more in the next few weeks, so we'll be talking more about that. Okay, thank you, everyone. See you next week.
Yeah. Bye, everybody.
Bye.
Right.